you see the slide?
Yes, clearly.
Okay.
Doctor,
So we'll start, okay? Because this is a little important topic and a little long lecture also. So we'll start now.
Doctor,
Yes.
can you record the lecture and send it to Telegram?
Actually, I cannot record the lecture because it is difficult, okay? Because it have to be stored, converted, edited, too much this one, okay? And today I am having 3 lectures. Of the third year also I have to do, okay? So
what if we record it? Is it fine?
what do you think?
What if we record it? Is it fine, doctor?
No problem. You can record it. No problem for me.
Thank you.
Okay, shall we start? Okay. Okay, can you hear me? All of you?
Yes, doctor.
Yes, please.
Okay, so we'll start. Okay, now we'll start. Carcinogenesis. Okay, so this week we are seeing cancer. Okay, I have explained you before that there is a cancer. Okay, so carcinogenesis means formation. Okay, genesis means formation, and carcino means cancer. Okay, carcinoma. We have studied carcinoma. Carcinoma means cancer, and genesis means formation. So how the cancer forms in our body? Okay, how the cancer develops in our body? Today the topic is that. Okay, that is called as the carcinogenesis. How cancer forms in our body, or what is the causes of cancer, and how the cancer occurs in our body. So today we are going to see. Okay, so the basic cause of cancer is a genetic damage. Okay, what is a genetic damage? Genetic damage is a damage to the gene. Okay, gene is a part of a chromosome. Okay, chromosome has DNA. In each cell there is a nucleus. In the nucleus there is DNA. Okay, and in the DNA, the part of a DNA, small part of the DNA is called as a gene. Okay, so when that gene is damaged, that is the basic cause of cancer. Okay, so the basic cause of cancer is a genetic damage. The gene is damaged, which is present in the DNA. Now, the 4 important genes which are important, which causes cancer, these are the 4 important genes. Okay, first the genes that promote growth. Okay, in all of us there is a gene that promote the growth. Okay, that is called as the proto-oncogene. Okay, so when this gene is stimulated too much, what will do? It will cause excessive growth. Okay, so this gene is damaged, proto-oncogene. Then genes we have in our body that is called as that inhibit the growth. Okay, it doesn't allow, it apply brakes to the growth. That is called as the tumor suppressor genes. Okay, when this gene is inactivated, when this gene is damaged and it is inactivated, there is no one to inhibit the growth, no one to apply the brake. Okay, so what will happen? There will be excessive growth. Okay, then there are genes that regulate apoptosis. Apoptosis is a process. I know I have taken you the lecture has been taken in a previous block, apoptosis. So whenever the cells are damaged, the cells undergo apoptosis. So when this gene is damaged, apoptosis gene, the cells which are damaged, they are not undergoing apoptosis, so they will multiply and they will cause cancer. Secondly, there are genes which are involved in DNA repair. When the DNA is damaged, there are genes which are involved in DNA repair. So those genes, they repair the DNA and make the cell normal. So what happens if this gene is damaged? The cell which had a damaged DNA, they are not repaired, and those damaged cells, they will multiply and they will cause cancer. So these are the 4 genes which are damaged in proto-oncogenes, tumor suppressor genes, apoptotic genes, and the DNA repair gene. Okay, now. So carcinogenesis is a multi-step process. It is not one-step process. Okay, it is a multiple step process in which result from there are multiple genetic alteration. Means multiple genetic damage is there, multiple genes are damaged, and then it will give rise to cancer. Okay, so it is a multi-step process. So you can see this is a normal cell. First one mutation is there, one gene is damaged, so it will convert into this cell. Then another gene is damaged, then it will convert into cancer cell. then another gene is damaged, then this cancer cell will multiply and it will form a mass. Okay, so this is called as this will give rise to a mass which is called as the cancer. So you can see there are multiple genes are damaged, not one gene. Okay, multiple genes are damaged. So one gene is damaged, it will convert into this type of cell. Then another gene is damaged, it will convert into founding cancer cell. Then another gene is damaged. Mutation means gene is damaged. Okay, what do you mean by mutation? Gene is damaged. So another mutation, then it will form a mass, it will multiply, it will form cancer. Okay. So this is the this thing. See, this is a normal cell. Okay, the normal cell. What will happen? I told you there is the main cause of cancer is the gene damage, genetic damage. Genetic damage means DNA damage because genes are present on the DNA. Okay, so what will happen? The gene is damaged. That is the DNA is damaged. And how this DNA damage? We will see there are many agents which cause damage to the DNA. Like chemicals are there, radiation is there, viruses are there, bacteria are there. So many agents they damage the DNA. They cause genetic damage and they cause cancer. Okay, so the many agents they call DNA damage, and afterwards what will happen? There is a failure of the DNA repair. The DNA is not repaired. Okay, so what will happen? These, I told you, growth promoting oncogenes. The growth promoting oncogenes, they promote the growth. So what will they do? When the DNA is damaged, they will be activated. Okay, they will be activated too much. So there is activation of the growth promoting oncogenes. Okay, so what will happen? There will be excessive growth. Then there is tumor suppressor gene, which will suppress the growth. But what will happen to this gene? It is inactivated. There is no brakes to the growth. So what will happen if there is inactivation of tumor suppressor genes? Uncontrolled proliferation, uncontrolled multiplication of cells will be there. Finally, the genes that regulate the apoptosis. Apoptosis is the process in which the cells which are damaged, the genes which are damaged, those cells they undergo death by apoptosis. So what will happen? They are damaged, so they will be decreased apoptosis. Okay, so all these genes, it will lead to excessive growth, clonal expansion, and the tumor will develop. That is called the tumor progression. Now see here, this is the causes of neoplasia. Neoplasia means cancer. So what are the causes of neoplasia? I told you there are chemicals, there are viruses, there are radiation. Okay, then there are some hereditary causes from birth only. Some genes are damaged from birth only. So those are the genetic defects. So all this it will cause cancer. So all this, what it will do? It will convert proto-oncogene. Proto-oncogene means the gene that promote growth. It will convert into oncogene. Oncogene means it will this gene, when it is damaged and it is excessively activated, it is called as oncogene. And this oncogene will cause cancer because this gene is excessively activated, and this will convert into oncogene, and it will cause excessive proliferation of the cells, excessive multiplication of the cells, and it will cause cancer. So the proto-oncogene will be converted into oncogene by radiation, chemical, viruses. Okay. Now see, what are there are some changes in the DNA, in the genes which cause cancer. You can remember this one, translocation. Translocation means, one part of the chromosome, it breaks and it attaches to the another chromosome. Like 22 chromosome, it breaks small part and it attaches to the 9 chromosome. So it happens in CML, that is the leukemia, chronic myeloid leukemia. Then 8-14 translocation in Burkitt's lymphoma, 14-18 in follicular lymphoma. Okay. Then deletion. In some chromosome, the part of the chromosome is deleted, like 13 chromosome is deleted and 17p chromosome is deleted. So it will cause retinoblastoma, which is tumor of the eye, colon cancer, tumor of the colon. Then sometimes the gene is amplificated. Amplification means the gene is multiplied. Okay, and that is happen in neuroblastoma and breast cancer. And sometimes there is an extra chromosome in the body, like trisomy 21, that is called the Down syndrome, extra chromosome in the body, and that will cause leukemia. So all these are the genes which are damaged, the chromosomes which are damaged. Now first we will see chemicals which cause cancer. Okay, I told you there are 3 important causes of cancer agent: chemical, viruses, and radiation. So first we will see how the chemicals. Okay, so chemicals are natural or synthetic, means they are natural chemicals also, and synthetic means artificial, which can cause cancer. And there is directly reacting or indirectly reacting. What do you mean by directly reacting? Directly reacting means directly they cause cancer. They do not undergo any conversion. They directly cause cancer. But but indirect means, indirect, they will, in the body, they go and they get converted into some other compound, which causes cancer, and that is carcinogenic. So the indirect chemical, they are called as procarcinogens. Okay, they go into the body and then they convert themselves into some other which cause cancer. Okay, so these are indirectly acting, and directly acting, they directly cause cancer. So what are the directly acting? Directly acting chemicals, they require no metabolic conversion. They directly cause cancer. So which are those drugs? Like drugs, a lot of drugs are there in the medical field, like alkylating agents. Okay, so these chemotherapeutic drugs are there, which are called as alkylating agents. If the patient is taking these drugs, it can directly cause, it will directly cause the cancer. Okay, so these are directly acting. Then procarcinogen, this is the indirectly acting. Indirectly acting, it is called the procarcinogen. It is not itself carcinogenic. It doesn't directly itself cause cancer, but in the body, it undergoes metabolic conversion in the body, and then it forms a compound which is called as a carcinogen, and this causes cancer. So here you can see procarcinogens. These are the indirectly acting chemical. Polycyclic aromatic hydrocarbon and the azo dyes. What will happen? These cigarette smoke or boiled or smoked meat, they go into the body, they convert into some other compound, and they will cause lung cancer and bladder cancer. Okay. Then finally nitrites. Nitrites, they are used as food preservatives. Okay. They go into the body, they are converted into nitrosamines, and they cause stomach cancer. Okay. Then aflatoxin. This is a toxin which is produced by the fungus. It goes into the body and it causes liver cancer. Then asbestos. Asbestos is a material which causes lung cancer and mesotheliomas. Mesotheliomas are the cancer of the pleura. So asbestos causes mesotheliomas and lung cancer. So these are all the procarcinogens, means the chemicals which go into the body and convert into another compound and they form cancer. Then what are the viruses which cause cancer? Viruses, what does virus do? Virus, they insert their nucleic acid. They insert their DNA into the host cell, and because of their insertion, they cause mutation. They cause damage to the proto-oncogenes, the tumor suppressor genes, and the genes regulating the DNA repair. Okay, so what the viruses do? They insert their nucleic acid. They insert their DNA into the host DNA, into the human DNA, and finally, because of that, what will happen? The proto-oncogene, the tumor suppressor gene, and the genes that regulate the DNA repair, they will be damaged. Okay, and there is a mutation. When they are damaged, what will happen? There is excessive cell growth, upregulation of cell division. Lot of cells will multiply, and it will lead to cancer. Which are the viruses which cause cancer? First, there are DNA viruses and RNA viruses. Okay, so DNA viruses are the human papilloma virus which causes cervical carcinoma. You have learned in your PBL. Okay, there is a cervical neoplasia which is caused by human papilloma virus. Okay, also it causes cervical cancer. It is caused by human papilloma virus. Then Epstein-Barr virus, it causes Burkitt's lymphoma and nasopharyngeal carcinoma. Then hepatitis B virus, it causes liver cancer or hepatocellular cancer. And herpes virus, it causes Kaposi sarcoma. It is a type of skin cancer. Okay, skin cancer. So these are the viruses which causes cancer. Oncogenic means onco means cancer. So these are oncogenic means causing cancer. So these are the DNA viruses which cause cancer. Oncogenic virus means the viruses which cause cancer. Then RNA oncogenic viruses. There is a human T-cell leukemia virus which causes leukemia and lymphoma. Leukemia is a cancer of the blood cells, and lymphoma is a cancer of the lymphoid cells. So this is caused by the human T-cell leukemia virus. Then there is also hepatitis C virus. It also causes liver cancer. Okay, it causes liver cancer. Hepatitis, it causes liver cancer. Then one bacteria which is called the Helicobacteria pylori. It is present in the stomach. Okay, and it causes stomach carcinoma and stomach lymphoma. So it causes stomach cancer. And the bacteria Helicobacteria pylori, it causes stomach cancer. Okay. Then radiation. The third important cause is the radiation. I told you these are three important causes. Chemicals, viruses, and radiation. So we have finished with chemicals, we have finished with viruses. Now we will see radiation. Radiation also causes cancer. And which are those important radiation? The ultraviolet rays. Ultraviolet rays of sunlight. Okay, they are very they cause skin cancer. Okay. Then X-rays. Okay, X-rays which we use for diagnosis. Then nuclear radiation, whenever there is nuclear radiation, lot of cancer can develop. And therapeutic radiation. Okay, so radiation, what it does, it damages the DNA, it damages the genes, and it causes mutation in the genes, the tumor suppressor gene, it causes damage to the genes, and it will lead to cancer. So you can see the ultraviolet rays. First, the ultraviolet rays. What it does, it damages the DNA. It causes pyrimidine dimers in the DNA. Okay, it damages the DNA and it causes especially the skin cancer. You know, squamous cell and the basal cell carcinoma and the malignant melanoma. These are all skin cancer, cancer of the skin. So it causes cancer of the skin. Then ionizing radiation, the radiation which we use like X-rays, gamma rays. Okay, these ionizing radiation, they also cause breakage of the DNA. Okay, they cause damage to the DNA. They break the DNA. Okay, and they cause leukemia. Leukemia is the cancer of the blood cells. Thyroid cancer and breast cancer and lung cancer. So X-ray, ionizing radiation, they cause leukemia, blood cancer, thyroid cancer, breast and the lung cancer. Whereas ultraviolet rays, they cause skin cancer. They cause damage to the DNA. Then you can see like this, the skin, you know, the ultraviolet rays, see, this is how the ultraviolet rays, it will first cause it will affect the skin, it will cause the skin cell, it will initiate the DNA damage, then this cell will be converted into one cell which is called the initiated cell. Okay, afterwards, again, this cell will multiply little and these are called the pro-neoplastic cell. Then after that, these cell will multiply excessively and they will form neoplastic cell and they will cause cancer. This is called as progression. So first there is initiation when there is a DNA damage, then there is promotion when those cells are promoted and they will form some few cells. After which these few cells will multiply too much, that is called as the progression and it will form cancer. Now see, these are the, so you can see these, we have already covered this. These are the three important environmental agents which cause cancer: chemicals, viruses and radiation. Okay, and this they cause damage to the DNA, because of which the proto-oncogene will be converted into oncogene and oncogene will cause cancer, excessive cell proliferation. So what is the proto-oncogene? Proto-oncogene is a gene which causes growth of the cell. Okay, it causes growth of the cell. So this is called as the proto-oncogene. Okay, so what happens to the proto-oncogene? You know, proto-oncogene can happen. There may be point mutation. How the proto-oncogene is converted into oncogene. Okay, so there is a point mutation, there is a gene amplification and translocation. I will explain you all these terms like this in this diagram. You can see this is the proto-oncogene. Okay, this is the proto-oncogene. So how it is damaged? First, there is translocation. Translocation means, you know, some other part of chromosome, it comes and attaches, some other gene comes and it attaches to it. You can see this orange yellow color. It will come here and what will happen? It will attach to this proto-oncogene and it will stimulate it. It will activate it. So when it is activated, it will cause excessive cell growth and it will cause cancer. So this is called as a translocation. Some new chromosome, it will come and attach to this chromosome, proto-oncogene, and some new gene will come in and attach to the gene and it will cause it activation. So this is translocation. Then gene amplification. Amplification means multiplication. This proto-oncogene only will multiply. It will develop multiple copies. So when it develops multiple copies, what will happen? It will cause excessive cell growth and it will cause cancer. Then thirdly, it will cause point mutation. Point mutation means one of the gene in the proto-oncogene will be damaged, and that will cause this whole gene to develop into oncogene. And this oncogene will cause excessive proliferation of the cells and cause cancer. So this is the same thing. This is the proto-oncogene which is converted into oncogene RAS, and it will cause, you know, colon cancer, leukemia. So this is the oncogene. Okay. Then see, this is the difference between the oncogene, and then finally there is a tumor suppressor gene. I told you oncogene, they cause excessive cell growth. And what is the tumor suppressor gene? It applies like brakes. You know, we have brakes to the car to stop it. So if there are no brakes, what will happen? You know, the cell will proliferate excessively. There is no one to apply brake to the cell. So the tumor suppressor gene, every one of us have normal tumor suppressor gene which control the growth. But if this tumor suppressor gene is damaged and it is inactivated, what will happen? The cells will proliferate too much. So this is the tumor suppressor genes. If the tumor suppressor genes are damaged, it will cause excessive proliferation of the cells. Okay, so which are the two tumor suppressor genes in our body? See, this is the two tumor suppressor genes which are there in the body, P53 and RB. These are very important. Okay, the RB is called the governor of the cell cycle. It governs the cell cycle. If the cell is multiplying too much, it will control. That's why it is called as the governor of the cell cycle. And P53 is called as the guardian of the genome. It guards the genome. It doesn't allow the cell to proliferate too much. So if these are damaged, the cell will proliferate. Okay. And P53 gene, this is a tumor suppressor gene, and it is called the guardian of the genome. Okay, it is called the guardian of the genome. What is does when the DNA is damaged, when the cell's DNA is damaged, what is that? It causes it repairs the DNA. It acts like a mechanic. Whenever something is damaged in the cell, okay, the DNA is damaged, so it will repair the DNA and it will make the cell normal. Okay, so this is very important. That's why it is called a guardian. Guardian means like a father. So guardian means guardian of the genome. It guards the DNA. If the DNA is damaged, it will repair the DNA. Okay, so what will happen if this gene is damaged? If P53 gene is damaged, what will happen? If the DNA is damaged, it will not be repaired, and the damaged DNA will continue to the damaged DNA cell will continue to proliferate, and it will give rise to cancer. So P53 is very important to repair the damaged DNA. So if this gene is damaged, the damaged DNA is not repaired, and the damaged DNA will give rise to abnormal cells, and it will lead to cancer. So this is the oncogene. Oncogene, this is I told you, proto-oncogene will convert into oncogene. These are the RAS, the APC, the FAP. These are all the oncogene. But tumor suppressor gene, these are the tumor suppressor gene. The P53 and the RB. P53 repairs the DNA, and RB, it controls the cell cycle. So if these tumor suppressor genes are damaged, there is no breaks to the cell proliferation, cell growth, and the cell will multiply excessively. This is a multi-step process. Okay, this is a multi-step. Carcinogenesis is a multi-step process. It is not one process. See, you can see from here there are accumulation of multiple mutation. Multiple mutation means multiple genes are damaged. Okay, not one gene is damaged. Multiple genes are damaged, and then it will lead to cancer. So here you can see this is how the carcinoma, the cancer of the colon develops. Okay, so see, this is the normal colon. Okay, this is the normal colon. This is the normal epithelium, mucosa, submucosa. Okay, first one gene is damaged. This APC gene is damaged. You can see this APC gene is damaged. So what will happen? The mucosa becomes risky. This mucosa becomes little damaged. Okay. Afterward, this proto-oncogene, that is the KRAS, it is damaged. Okay, and it will convert into oncogene. Okay. So what will happen? Finally, it will proliferate and it will form an adenoma. You can see adenoma. Then after this, the P53, that is the guardian of the genome, this is damaged. Then this will convert into carcinoma, that is the colon carcinoma. So this is how it develops. First there will be hyperplasia, then there will be adenoma, there will be dysplasia and carcinoma. So all these changes take place when there are multiple mutations. One gene is damaged, it will cause hyperplasia. Another gene is damaged, it will cause adenoma. Then another gene is damaged, it will cause dysplasia. Then again P53 will be damaged, it will cause cancer. So this is how the colon cancer, it is a multi-step process. Carcinogenesis is a multiple process. Now DNA repair gene. Okay, I told you when the DNA, there are some genes will repair the DNA. Okay, so because if the DNA is damaged, it will lead to malignancy. So the DNA have to be repaired if it is damaged. So how the DNA is repaired? You can see here there is first, suppose this the DNA is damaged, this red area, here the DNA is damaged. So what our body does, it will remove this one, it will remove this one and it will repair it. Then suppose there is mismatch repair, like adenosine, guanosine, there are pairs. So if there are mismatch repair, faulty pair, so what will happen? The body will repair it, it will cut it and it will repair it. Okay. Then finally, sometimes there are two pairs. It will be fused, like it will call thymine dimer. Thymine dimer. It will be fused. So this is also DNA damage. So it will be removed in our body. The repair process, it will remove this part and it will repair it. So these are the DNA repair gene. It will repair the DNA. When the repair gene is damaged, that time also the damaged DNA will give rise to cancer. Now, secondly, the apoptotic gene. Okay, the apoptotic gene is there. I told you when the DNA is damaged, those cells undergo apoptosis because they are defective cells. And that is caused by apoptotic gene. So if the apoptotic gene is damaged, there will be no apoptosis, and those damaged cell will continue to proliferate and it will cause cancer. So apoptotic gene is also very important. So you can see apoptosis role in cancer. If there is no apoptosis, the abnormal cell will proliferate, it will multiply, and it will cause cancer. So what happens the cells to be malignant? How the cells become malignant? Because I told you there are four important genes which are important. First, the tumor suppressor gene, the proto-oncogene, the DNA repair gene, okay, and the apoptotic gene. So all these four genes are, if they are damaged, they will give rise to carcinogenesis. Okay, so it is carcinogenesis is a multi-step process. What do you mean by multi-step process? There are multiple mutation, multiple genes are damaged. So if one gene is damaged, there is initiation. Okay, then after second gene is damaged, there is promotion, then there is transformation, there is progression, and there is invasion and metastasis. We will see in the following picture, we will see like this. Okay, so what is initiation? Initiation, like some chemical, they damage the DNA, they damage one gene. Okay, they damage one gene, and that is called as the initiation. So they damage one gene, and that cell is converted into some abnormal cell, that is called as the initiation. Okay, that is irreversible. Now promotion, the another gene is damaged after exposure to the initiation. After that agent is continuously exposed to that, exposing to that cell, what will happen? Another gene is damaged, and it will cause promotion. It will allow that cell to multiply little. That is called as the promotion. Finally, progression. If the genes are again damaged, many genes are damaged, the tumor will multiply and it will progress to a large mass. So that is called as the progression. So here you can see this is the normal cell. Okay, one gene is damaged by the chemical, it will convert into tumor cell. Then after that, another gene is damaged, it will multiply little, that is called as the promotion. You can see this is the promotion area. Finally, if many genes are damaged, it will multiply and it will form a mass, that is called as, this process is called the progression. So first there is initiation, then there is promotion, and then finally progression. In progression, the cell multiply very fast. So here you can see, this is a good picture. Here you can see first this is the normal cell. Okay, the carcinogen, that is the chemical. Okay, it will attack it and it will convert into this type of cell. It will convert into sometime abnormal. It will damage the DNA and it will convert into this type of cell. This process is called as the initiation. You can see this is the initiation. This is the initiation process. Okay, then what will happen after this? Another gene is damaged. Okay, another gene is damaged, and this cell will multiply little. Okay, it will multiply, it will undergo hyperplasia. Okay, it will undergo hyperplasia and it will multiply little, and it will convert into, and it will continuous exposure of that chemical, it will lead to hyperplasia, and the cell will multiply, but it will not multiply, it will multiply little. That is called as a promotion. Okay, promotion. Finally, what will happen if the chemical is continuously exposed? Many genes are damaged, and finally these cells will multiply excessively, and they will form many cancerous cells. That is called as a progression. So you can see this progression where the cells are becoming abnormal, dysplastic, and they are multiplying very much. So this is the progression. After progression, what will happen? Invasion means these cells, they will invade the blood vessels. They will go into the blood vessels and the lymphatics, and they will go into different organs like the liver, the lung. Okay, and that is called as finally invasion. You know what do you mean by invasion? These cells they will multiply too much. Okay, they will multiply very much, and they will invade the surrounding organs. Cervical cancer. If invasion means it will spread to the surrounding organs. What is surrounding the cervix? It is the urinary bladder. It is the rectum. Okay, so these are the surrounding organs to the cervix. So if that is called as the invasion, it will invade the surrounding organs. Like if cervical carcinoma is there, it will invade the rectum. It will invade the urinary bladder, the urethra, the surrounding organ. That is called as the invasion. Okay. After invasion, what will happen? These cells will go into the blood. It will go into the lymphatics, and this will go through the blood to the different organs which are far from the area, from like the liver and the lung and the bones. That is called as the metastasis. You will learn in your coming lecture how the cancer spreads. That is called as the metastasis. To the blood, and from the blood it will go to different organs like the liver, the lung. Okay, like cervical carcinoma, it will go to the lung, the liver, and that is through the blood. The malignant cell will enter the blood, and that is called as the metastasis. Okay, so this is how the cancer is there. First is the initiation. The cell is converted into an abnormal cell. After which the cell is another gene is damaged, the cell will multiply and it will cause hyperplasia. It will cause promotion. Finally, the cell will multiply too much. Many genes are damaged, and they will convert into dysplastic cell. They will multiply excessively. That is called as a progression. After progression, what will happen? They are multiplying continuously, so they will invade the surrounding organs. The surrounding organ, that is called as the invasion. After the surrounding organ, the malignant cell will enter the blood, and they will go to the organ which are far, like the liver and the lung, and that is called as the metastasis. Okay, metastasis is when the cell goes to different organs. Okay. So this is called as the metastasis. So here you can see this is how the causes of neoplasia. This is the summary. Okay, first there are environmental agents. Environmental agents are which agents? Environmental agents are chemicals, viruses, radiation. Okay, these are all called as carcinogens. Carcinogens are the agent which cause cancer. So these are chemicals, viruses, and radiation. So these are the environmental agents. So what it does, first it will attack the proto-oncogene and it will cause proto-oncogene to activate it. It causes activating of the proto-oncogene. So this proto-oncogene will be activated, because of which it will convert into oncogene, and this oncogene will cause the cell will multiply excessively. Okay, and it will cause cancer. Okay, so this proto-oncogene is damaged. Secondly, which gene is damaged? The tumor suppressor gene. This tumor suppressor gene usually suppresses the growth. So it causes tumor gene inactivation. This is inactivated. So there are no breaks to the okay, there are no breaks to the cell. Okay, and it will cause, so this tumor suppressor gene. Okay, and finally, which gene is damaged? The apoptotic gene and the DNA repair gene. All these 4 genes will cause cancer. Okay, so this is how the cancer develops in our body, and this is called as the carcinogenesis. So I hope all of you have understood what is carcinogenesis. Okay, are you getting it? Okay, let me take the attendance. Okay, don't go. Okay, all of you have understood? All of you have understood or no?
Yes, doctor.
Why you are not saying? Okay, all of you, are you with me?
Yes.
Okay, can you hear me, all of you? Okay, all of you listen here. Any question you want to ask?
Thank you.
1 minute wait. Okay, let me take the attendance. Any question you want to ask? Okay. Can you, all of you can hear me? All of you can hear me or no?
Yes, doctor, we can hear
Yes,
you.
doctor.
Okay, you can see this picture now. Last time I told you benign and malignant tumor. Can you see this picture?
Yes, endometrium.
So this, I wanted to explain you once more this one. This is very important, the difference between benign and malignant. So I just want to explain the benign tumor is a small mass and it is covered by capsule. It will not grow invade. This is invasion into the surrounding tissue. It will not go into the blood. Okay, and it is well differentiated. And whether the malignant tumor, it is large in size, irregular. It will spread, invade the surrounding tissue, and it will go into the blood. Okay. Last time I forgot to tell you this one, signs of malignancy. Okay, in the cells, you will see dysplasia. What do you mean by dysplasia? The cells become abnormal. Okay, if the cell becomes malignant, what you will see in the cells? Okay, so the first, what you will see in the cell and what you will see in the nucleus. So in the cell, you will see dysplasia. Dysplasia means the cell will become abnormal in shape. Disorganization means the cell will become disorganized. They will be arranged anyhow. Loss of polarity also means the same. That cells will not be arranged in a uniform manner. It will be arranged in anyhow. Pleomorphism means variable in size and shape. Okay, the cell will appear in many cells will appear in variable shape and size. Loss of cohesion. Loss of cohesion means the cells will detach from each other. Okay, they will be separated from each other. And the giant cell you will see. So if you see all these features, these are the signs of malignancy. In the nucleus, what you will see, the nucleus will become large. The nucleus will become large in size. If the cell becomes malignant, the nucleus becomes large in size. Then the nucleus becomes dark in color. That is called the hypochromasia. The nucleus will become dark in color. Okay, the nucleus becomes high. That's why it is called the nucleus cytoplasmic ratio will be high. Okay, pleomorphism means the nucleus size will be variable in shape. There will be various size nucleus. The nucleoli will be prominent. You will see the nucleoli in the cell. Okay, and there will be a lot of mitosis, abnormal mitotic figures and mitosis. So these are all the signs which you will see in the malignant cell. Okay, and finally the cell will lose its function, and there will be loss of the normal function of the cell. That time it is called as a malignant cell. Okay, so this is the summary last time. Neoplasia, it is an uncontrolled cell division which is not responsive to the growth control, and benign tumors will end with OMO, and malignant cell will end with carcinoma and sarcoma. Benign tumors are slow growing, they are well differentiated, and they are localized, they do not metastasize, and they will be easily resected by surgery. And malignant neoplasm, they tend to be very fast growing and they invade the normal structures. So malignant neoplasm will be poorly differentiated, whereas the benign will be well differentiated. And malignant neoplasm will invade and infiltrate into the surrounding tissue, and they will undergo metastasis to the lungs, liver, and the bone and brain. Okay, so I hope all of you have understood. Any questions? Any question you want to ask? Okay, so then I will end the meeting. Okay, assalamu alaikum warahmatullahi wabarakatuh.